study about the determination of packed cell volume or hematocrit. That is the solid part which is present in our plasma. We will study about that volume. So what is packed cell volume or hematocrit? It is defined as the volume of red cells in a given sample of blood. It is expressed as a percentage of the total volume of the blood sample, and it is done by centrifuging a column of blood in a glass tube. After the centrifugation, we can determine the packed cell volume.
Expressed as percentage of total volume of the blood sample. Expressed as percentage. Percentage of what?
Percentage of RBCs. Percentage of RBCs.
RBCs. Percentage of RBCs. Okay. And I have another question. Let it be finished, then I will ask.
So, in order to standardize the degree of packing during centrifugation, there are some minimum necessary steps we have to follow. That is, there should be optimum speed of the centrifugation machine. There should be optimum radius of the centrifuge, and there should be an optimum duration of the centrifugation. Can I change the slide?
Yes, sir.
So the determination of PCV is simple. We will do it with a machine, with a centrifuge machine. So it is a simple technique. It is very accurate, and by this we can know whether the patient is anemic or he is having increased mass of RBC in his blood. So these are the pipettes we will be using to draw the blood. This is the image of the centrifugation machine, which is present over here.
It is a better version of Pranava. This one is the centrifugation. Now, what you have come here to look inside the, see, the image that is shown here. Similarly, the centrifuge machine looks like that from inside. Okay. Okay, now you can see one by one.
Once the class is over, you can see it. Okay, go and see. Go and see. Everyone go and see the machine.
Whoever wants to see, just see, okay? Centrifugal force comes into action. Motion, which motion? Revolutions. Okay, it is like revolving around something. Okay. Have you heard of this? To be देखा है ना ऐसे extreme में at the end it bond around this one can. Okay. The force that's acting outward is centrifugal. Fugal not be there. Capital is inside one. Centrifugal. What's the capital is centrifugal.
So these are the tubes in which we will keep the blood and we will centrifuge. So how will we determine PCV? So firstly we have to add anticoagulant to the blood so that the blood doesn't coagulate. We will be adding calcium oxalate with the blood, and we will use a hematocrit tube or Wintrobe's tube. In our case, we will be using an Wintrobe's tube, and there should be a specific rate and revolution. I already told you for the centrifuge machine. So it would be spined at 3000 RPM at a duration of 30 minutes. For 30 minutes, we have to centrifuge the blood at 3000 RPMs. So after centrifugation for 30 minutes, 3 layers will be separated. One would be the superficial layer, which will be containing the plasma. Middle one will be a thin layer, which will contain which will be the buffy coat, which will contain WBC and platelets, and the lowermost layer will contain the packed cells, the packed RBCs. So this is the same thing. They have shown it in an Wintrobe's tube. There is the first layer is the plasma layer, then there is the buffy coat, and then the last layer is the packed RBCs or the hematocrit. So there are several methods. One is macro method using the Wintrobe's tube. One is micro method using the capillary tube. and third one is automated analyzer. In our class today we are going to do the first one, the macro method using the Wintrobe tube. Here you can see there are two Wintrobe tubes.
Okay. सामने से देखो.
It is graduated from 0 to 10. See the graduations.
এবার তুমি মনে করো এইটুকু পেক্সেল ভলিউম দিস মাস রিডিংটা কত হবে? হ্যাঁ। এটা পুরো ঠিক মতো ক্যালকুলেট করতে পারছি না রিডিং
Suppose the calibration is from 0 to 10, na? You are saying from 0 to 10, right? Suppose till 4.2 marks, there is the volume of RBCs is still 4.2. Can you tell me what is the PCV of the blood or hematocrit?
42%.
Yes. Right. It will be out of 10. We are taking sample till 10. Okay. 10 में 4.2 is the RBC volume. Okay. So the total PCV will be 42%. 10 में 4.2, 100 में कितना होगा? 42.
You can see that the base of the tube is flat, and there are two types of markings. One is going from below upwards 0 to 10, and another is going from upwards to downwards 0 to 10. So which one would you consider while you are?
Bottom to top, 0 to 10.
Everyone knows what we will consider while measuring the PCV.
So the white part is
hemoglobin measure
No.
You will use the markings from downwards to upwards, from 0 to 10. Where it is written that below one is 0 and upwards is 10, you will go in this direction. The second one is used for ESR counting, for counting the ESR. This one is used for counting the PCV, for calculating the PCV.
You will use 0 to 100, so 0 is at the bottom. Another scale is there, 0 is at the top. So which one you are going to use for this?
So I have already told you that we will be using anticoagulated blood sample for this test. So which anticoagulants we are using? One is DR.TA, heparin or double oxalate. And the specimen doesn't need to be a fasting sample. What do you mean by a fasting sample?
12 hours fasting.
Okay, you can eat and give blood also. That will not affect the PCV. So these are the anticoagulants used.
The specimen need not be a fasting sample.
Fasting sample. So they can eat and give blood also.
They can.
So what is the principle on which this is acting, or how you are calculating the PCV? When anticoagulated blood is centrifuged in a hematocrit tube at high speeds, the erythrocytes sediment at the bottom. The red cell column is called a pexel volume. So this is the simple thing. When we are centrifuging the blood, the PCV, as they are the red blood cells, as they are having a higher mass, so they will settle down. So this column we will use for calculation of the PCV. Then what are the requirements whenever we are doing the experiment? So which are the things we will require to do the PCV? So one is the specimen, obviously, the blood which we will be giving. There should be an anticoagulant blood, anticoagulated blood. Coagulated blood we cannot use. Then there should be an Wintrobe tube, a pasteur pipette, or a syringe with needle, and a centrifuge machine. In this case, we have to consider that whenever we are taking the blood and whenever we are pouring the blood into the Wintrobe tube, we shouldn't use any needle. Otherwise, what will occur if we are using the needle? Coagulation.
Some blood will be left in the tube.
No, suppose we are taking out the blood with a syringe. Then obviously in a syringe there will be needle. But we will not use the needle to pour the blood into the Wintrobe tube. Why?
We basically remove the needle part, and so the syringe only we just
Not have the
pour the
contamination.
blood, removing the needle part. Why?
But we have already taken with the needle. Na? In this case, we will not use the needle because if we are using the needle, there will be a compression, due to which there may be hemolysis or fragmentation of the RBCs. So this will give a false result. So that's why we will discard the needle
Basically, what we do is we use a needle with a larger bore. Okay, now the needles that are being used in the lab basically have a very smaller bore. The diameter is very less, the inner diameter. So once you have taken blood, but once you are pouring the blood in the
Prevent hemolysis.
or the capillary tube, sorry, not capillary tube, Wintrobe tube or the penicillin tube. In the first time only you will see in the process there is some tube called penicillin. So once you are pouring the blood, if the bore is very less, what will happen is RBCs will get hemolysis. There can be lysis because of pressure of the flow to the needle. So either you use a needle with a larger bore, or you just take out the needle and through the syringe only you just put the blood. Okay.
Then the diameters,
Use a large needle with a larger bore. Diameter is bore in case of a larger bore.
then what are the specifications of the Wintrobe tube? Second is the glass tube is 11 centimeter long, but there are markings up to 10 centimeter. You have seen that there is marking up to 10 centimeter, but the whole glass tube is 11 centimeter long. The internal diameter is 3 millimeter, and the base is a flat base. The inner base is a flat one. The tube is calibrated as all of you have seen. There is calibration from 0 to 10 and from 10 to 0. Markings are there, no?
Yes.
One for ESR and one for PCV. There are two kinds of markings in this tube. So these are the calibrations. Then it holds in total of 1 ml of blood.
Important diameter, length, how much blood it can contain.
Now the procedure. How will you do the PCV? How will you count the PCV? So first we will mix the blood sample carefully. We will mix the anticoagulants with the blood. Then we will prepare the blood for the PCV counting. Then we will label a Wintrobe tube. We will fill the tube using a pasture pipette or a syringe up to the 100 mark. I have told you that we would use a wide bore, or you may, in case of our lab, if you are not getting a needle with wide bore, we will discard the syringe and we will just use the remaining part of the needle to pour the blood. Otherwise, there might be hemolysis or breakdown of the blood cells. Then avoid trapping of air bubbles. Then what we will do? We will pour the blood into the pasture pipette or this Wintrobe tube. Then we will place the tube in a centrifuge cup. We will use another Wintrobe tube filled with another specimen to balance the weight for centrifugation. We will centrifuge at 3000 RPM for 30 minutes.
In 1 minute, it will revolve how many times?
3000.
3,000. What would happen if I revolve it in more speed, say 8,000?
Hemolysis.
And what would happen instead of 3,000 I use 2,000 RPM?
It would not settle properly.
The separation you will not be able to separate.
Now 3000 RPM for 30 minutes. Per minute only.
This is per minute. Revolutions per minute.
For
This speed. At this speed it will run. How long? For 30 minutes.
So after the blood is centrifuged, there will be 3 different layers. One is the plasma, one is buffy coat, and the pack cells. So these are the 3 layers which will be seen. The upper part is plasma, which will be pale yellow in color. Thin layer of WBC platelets, which is called the buffy coat. It will be in the middle one, and the lowermost layer will be of the red blood cells. So these things already I have discussed.
Buffy coat, how much it will be?
1 millimeter.
Length? 0.5 to?
just note that down.
0.5 to 1 mm. How many cells would be there approximately?
Then if the color is reddish, it may indicate hemolysis. Because in hemolysis, the red cells will be broken down, so they will have a lighter weight, so they will settle down in the topmost part. So reddish will indicate hemolysis. What are the other uses of the Wintrobe tube? So it is for ESR estimation. So I've told you that from 0 to 10, if you count downwards, it is used for ESR. The Westergaard method is preferred as it is more accurate. So some clinical conditions in which there will be fall in the hematocrit. So these are anemia, then hydremia, hemorrhage, cirrhosis, renal diseases, and in case of pregnancy also there will be fall in the hematocrit.
Yes. That's why during pregnancy PCV is less. That is the reason. Yes, ectopic pregnancy is also there, and false pregnancy is also there. Same, there will be increase in size of the uterus. From outside it will appear there is pregnancy. Blood test it will show. But there is no fetus. Molar pregnancy. This is called molar pregnancy. Hydrated mole, actually hydrated mole, grapes-like structures will be, a lot of grapes-like structures will be there.
So what is hydremia?
Hydremia is
Or more water content in people?
There are some conditions in which there will be decrease in the hematocrit. Sorry, increase in the hematocrit. So it is, first one is polycythemia. Then there will be increase in dehydration.
dehydration. Why dehydration will increase the hematocrit?
Because plasma content gets reduced.
Plasma, water that is dehydrated. So it is becoming concentrated. And polycythemia? What is polycythemia?
Excess production of RBCs.
So there is, proportionately there will be more RBCs.
Then emphysema.
Emphysema, what happens? What is emphysema?
The alveoli walls are damaged. Alveoli walls.
Damage becomes fused.
Fused.
Yeah. Becomes fused. As a result, what happens?
Surface area decreases.
Hypoventilated. Not properly ventilated. So as a result, what happens? If not properly ventilated, what happens? What will happen? Sufficient oxygen will not be entering into blood. So what will be there? Will be hypoxia. So this will stimulate erythropoietin. So it will increase RBC formation.
Then comes congenital heart disease. In congenital heart disease, the person will have a diseased heart even from the beginning of his birth. So there will be reduced oxygen level in his blood. To compensate this, body will increase the RBC count. So in the similar way as emphysema, due to hypoxia, there will be increase in hematocrit. Then extreme physical exercise. In people who are doing extreme physical exercise or very high physical activities, they will be having more requirement of oxygen in their muscles. To fulfill this requirement, body also has to increase the concentration of RBCs in the blood, in the plasma, in the blood. So that's why hematocrit will also be increased in people who are doing extreme physical exercise or heavy works.
Okay, then next.
Then what are the normal ranges in the different age groups and different genders?
Important.
For men, it is 42 to 52%. For women, it is 36 to 48%.
Why it is less in women?
Less muscle.
Why hemoglobin is less in women? Why RBC count is less in women? Why blood volume is less in
Because
women?
of less weight, the requirement of oxygen is less.
Because of testosterone. Testosterone because of the directly linked to the for production.
সবগুলো ক্লিন করা হয়েছে এসিড দিয়ে দেখে করো
Don't try to use mobile device. During exam, don't try to use mobile. Don't try to use mobile during exam.
देखो कुछ लाइन very faintly nice to see some lines. Paper is better to focus on some other lines. Maybe
Forget this.
how much minutes?
During exam, some of your students they use mobile. Right. And the company exam. Whoever